Hi, so this is the second half of part 1 of chapter 3. Most of this is going to be related to membrane transport. So I'm going to discuss how substances are moving across the plasma membrane of a cell. Like I said, most of the rest of this PowerPoint has to do with that topic. Related to this topic, I did make a document for you all called Diffusion and Osmosis that I posted yesterday, I believe. Please take a look at that. I really think if you go through that, I've detailed everything out for you. So if you can go through that document and understand it, you will be able to answer any question about membrane transport. You will definitely have 2 or 3 questions on your next quiz about this. So make sure you understand that document. So this first part really relates to that document. Okay, so membrane transport. We've mentioned that the plasma membrane is selectively permeable, and you already know what that means. Some things will get through, others will not. You should know that there are 3 main types of membrane transport, and we'll talk about each of them. The first one is passive transport. It's called passive because it does not require energy to occur. It does not require ATP. So typically this is going to be due to just random molecular motion. And we're going to talk about specific examples of passive transport, such as filtration, diffusion, and osmosis. These are the 3 kinds of passive transport that you need to know about. But for this course, we're really going to focus much more on diffusion and osmosis. In Bio 139, you will go in detail about filtration. So we're really not going to cover the details of filtration, mainly diffusion and osmosis. Second type of membrane transport is called active. And it's called active because it does require energy. It does require ATP. And we'll talk about a really important example of active transport as a type of pump. It's called the sodium-potassium pump. That is a really important pump in cellular physiology. So definitely we'll talk about it. Also, another kind of active transport is vesicular transport. So we'll mention that. So active transport, no matter what, requires energy in the form of ATP. The third type of membrane transport is called carrier-mediated transport. So this occurs when, let's say some sort of substance is not able to get through the membrane by itself. So it's going to need some kind of help in getting across. And so what we'll see is that there will be a memprotein, such as a channel, for example, or a carrier that is going to transport that substance across the membrane. And it says here it's typically sort of like a passive process. It basically, carrier-mediated transport, most of the time, does not require ATP. So let's start with filtration. Again, I don't want to focus so much on filtration. You might just have one really basic question about it, if you get a question about it all, because you'll be focusing on this next semester. Just for us, what we really need is a definition. So filtration as a type of passive transport really is going to involve the movement of substances based on what is known as hydrostatic pressure. So that means the pressure that water exerts on the cell wall or the cell membrane is going to cause or drive the movement of substances. All right. So basically, filtration you can just define as the movement of particles based on hydrostatic, or we can say physical pressure. Specifically, it's hydrostatic pressure. It is water that's serving as this physical source of pressure on the plasma membrane, and this causes substances to move. Attached to this, really your greatest example in physiology of filtration is going to be in your capillaries. This is how substances such as solutes are going to pass through the walls of your capillaries is going to be based on filtration. The next picture is showing you a graphic of filtration. Again, really, I'm not going to ask you details about this at all. You can do that next semester. All you need to know for now is that filtration is when substances are driven into or out of the capillary wall or a blood vessel wall based on pressure, such as pressure coming from water. Now, this is important. So we're getting into simple diffusion. Really important for you all to understand simple diffusion, which relates very much to the document I made for you all. So let's talk about what this is. Simple diffusion involves when you have, let's say, for example, a cell, and that cell has a difference in concentration of a certain item. This could be an ion, it could be a substance. We're dealing, when we talk about simple diffusion, we're dealing with a solute. We're dealing with a solid. All right, so we're going to be dealing with the movement of solutes across the plasma membrane. So what ends up happening is, let's say we have collisions between molecules in areas of high concentration. Let's say we have a whole bunch of sodium inside the cell, for example. If we have a whole bunch of sodium or a high concentration of sodium inside the cell, and on the outside we have a low concentration of sodium, well, this means there's a difference. There's a difference in concentration of sodium on the inside versus the outside. When we have this difference in concentration of a solute between two areas, we can say that we have a concentration gradient. Gradient really just means, yes, there is a difference in the concentration. Difference is what this really means. So in simple diffusion, what's going to happen is that molecules, remember we're dealing with solutes here, solutes are going to move from whenever there's a difference, whenever there's a concentration gradient, that substance is going to move from where it exists in high concentration to where it exists in low concentration. Okay, that really is your definition here of simple diffusion. The movement of a solute from where it exists in high concentration to where it exists in low concentration. And this is just an effort to maintain homeostasis, to maintain balanced levels of this solute, whatever it may be, on both sides of the membrane. Remember, this is a passive kind of transport, so ATP or energy is not going to be required. Now, you can have different rates or speeds of diffusion, and this is going to be influenced by a number of variables. One thing that will affect how quickly diffusion happens is the size of the molecule, temperature, for example. And so actually that's what this slide has to do with. And I'm not sure why this slide shows, these are supposed to be arrows, but they're really not coming out. I think on your actual slides you see them, but here in Collaborate for some reason it doesn't show these arrows. All right, so but we can go through it. So passive transport, simple diffusion. This slide is telling you about the different variables that can influence how quickly diffusion happens or how slow diffusion happens. So let's take the first variable, temperature. So if you have high temperature, you will increase the rate of diffusion. Molecular weight, so basically how much does the molecule weigh? All right, what is its weight? So the larger molecules are going to move slower. So basically, if a molecule has a high molecular weight, it's slower to move. The smaller or the lower the weight of that molecule, the easier it is to move, the easier it is to travel. So low molecular weight, you would have increased diffusion, right? If you have high molecular weight, you're going to have slower diffusion. Steepness of the concentration gradient. So remember, gradient means difference. There's a difference in concentration of a solute on either side of the membrane. So on both sides of the membrane. So when you have this, if it's a really high difference, okay, the greater the difference in concentration, the greater there is a drive for the solute to move. So the higher that difference is, the higher the rate of diffusion. Membrane surface area. Okay, so we actually mentioned something called microvilli that increase the surface size of the cell. So that would be kind of an example of this. The greater surface area there is, okay, the higher the surface area, the greater the rate of diffusion. Just because there's more space there, there's more space allowing a greater number of molecules to move across that membrane. And of course, membrane permeability. So permeability, you remember, is referring to how, if it's easy or not easy for something to pass across the membrane. So if something is high in permeability, meaning it has a greater potential to offer solutes to cross it, then yes, there will be a higher rate of diffusion. So I'm sure you'll have a question about one of these variables, about how these variables affect the rate of diffusion, whether they will increase or decrease the rate. Passive transport, again, just going on about this. Okay, so molecules have a natural drive to diffuse down their concentration gradient. Okay, so when we say something's moving down its concentration gradient, what that means is that substance is moving from where it is in high concentration to where it exists in low concentration. It's moving down its concentration gradient. And so when we talk about this in terms of the plasma membrane, of course, we're talking about the intracellular fluid and the extracellular fluid, the concentration of that solute in both of these places. And again, so the plasma membrane actually helps create this difference because it's a barrier, right? It's a barrier between the ICF and ECF. So yeah, you will see differences in concentration of solutes on each side. Now this is important. This part here is important. You should definitely know, all right, that based on the structure of the plasma membrane, remember that all-important structure, how we have those phospholipids arranged in the bilayer. All right, we have, remember, polar heads, the ICF and ECF, and in the middle we have our fatty acid tails. Well, I think I mentioned this already, that nonpolar substances will diffuse directly through the plasma membrane. Nonpolar means they're lipid-based, so they're able to get through those fatty acid tails. So you should know that nonpolar substances use simple diffusion. They're able to directly pass through the plasma membrane to equal out the concentrations of it on either side of the membrane. Examples of nonpolar substances that you should know. Oxygen and carbon dioxide are nonpolar substances, right? So it's very important for these two gases to be able to easily go in and out of cells. We have to have that ability, all right? So you should definitely know these examples. Probably have a question about it. So oxygen, carbon dioxide, very important examples of nonpolar substances that use this simple diffusion. Fat-soluble vitamins, all right? So again, fat-soluble, that means it's nonpolar. So most small substances that are nonpolar go through this process of simple diffusion. They do not need any help getting across the membrane. They can go right through. All right, and that's kind of what's said here in this statement, all right? Not the nonpolar hydrophobic lipid core of the plasma membrane blocks diffusion of most other molecules. All right, so we're talking about, for example, hydrophilic molecules. Hydrophilic molecules, with the exception of water, cannot easily pass through this bilayer that you find in the plasma membrane. And so again, examples of molecules that can passively diffuse across the plasma membrane would be lipid-soluble and nonpolar substances. Now, some very small molecules are able to, but most of them are going to require some assistance through channels, okay, especially if these small molecules are hydrophilic. All right, and again, larger molecules, they're going to need assistance, all right? We're going to use carriers to transport larger molecules. Here's a picture of simple diffusion. Here's your plasma membrane there. And it's showing here soluble solutes, right? What we have here is, notice there are a lot more of these on the outside of the cell, the extracellular side, compared to a smaller number of these solutes on the inside of the cell, or the cytoplasm. So this means we have a concentration gradient. We have a difference in the concentration of solutes on either side of this membrane. So based on the laws of simple diffusion, if it's a lipid solute, it is simply going to pass through. All right, so we're going to have the movement of this solute from the outside to the inside to equal out or balance out the concentration of this solute on either side. This is just a natural drive that molecules have. All right, they're going to want to go where they exist in lower concentration. So osmosis is the second type of passive transport, actually third. We mentioned filtration, but for our purposes, we're really focusing on simple diffusion and osmosis. Pay special attention to osmosis because students do tend to have some misunderstandings about this. And some sources that you go to, like maybe your textbook or other books that you've read in the past on this, if you have, really complicate what's going on here. They make osmosis seem a lot more difficult than it really is. So I want to simplify it for you. And I did that in the document that I posted for you all. So please go through that and you will see there's a very simple way to look at this. First of all, first most important thing to know about osmosis is that it involves the movement of a solvent, not a solute. Okay, a solute or a solid is not moving in osmosis. All right, solute is moving in simple diffusion, which we just talked about. Osmosis is the movement of solvent. And in the human body, we're really talking about water. This is the main type of solvent that we're dealing with in the human body that will perform this behavior of osmosis. Okay, so now remember, the plasma membrane is going to allow non-polar substances to pass. But I mentioned to you last time that water, because of its really unique shape, because of its angles, it is able to finagle through those fatty acids. All right, but we do have this backup plan just in case we're not able to get that very hydrophilic water molecule across the membrane. We do have these special channels, and these channels you should definitely know the name of. They're called aquaporins. Aqua for water, from the Latin water. Aquaporins, all right, porins, like a pore in the membrane. Okay, so these are water channels. So just in case we can't get our water to pass through the membrane on its own, we're going to have these channels that will escort or transport this water through. Okay, so very similar to simple diffusion, osmosis is going to involve the movement of water based on a concentration level. I think what confuses students sometimes is they will try to remember, okay, water is going to move, just like in diffusion, water is going to move to where it exists in high concentration to where it exists in low concentration. So that's a similar definition of what simple diffusion is when it comes to solutes. But I think it's better for you to look at water movement based on not water concentration, but solute concentration. And initially that might not make sense. But if you think of it that way, it's going to help you with some other terms that you need to know. So I think it's a great way to look at osmosis in this way, that water is the greatest solvent we know. Water exists to dissolve. That's what water does. It dissolves solute. That is what it does. So what that means is that water follows solute. And if you just keep that in mind, it's really going to help you with understanding some other terms we're going to talk about. Water, because it likes to dissolve, is going to follow, or it's going to go towards wherever there is a high amount of solute. Okay, so try and remember it that way. And remember, solute is not moving in osmosis. Here is a little picture of osmosis. So here's our little plasma membrane here. And let's say, okay, out here we've got water on the outside, we've got water on the inside. And all I'll let you hear is that you can do both with water. So water can directly diffuse, okay, through the plasma membrane. Remember, water is an exception when it comes to polar substances that are able to pass this membrane. But if we can't get enough of that water in, we have this backup plan here. We have this aquaporin. It's a channel that will go ahead and transport these water molecules in. So clinically, all right, burn patients are an example of, if we have damage to this plasma membrane, then we lose this ability to have concentration gradients. And so this is going to make this person lose a lot of electrolytes, for example. They're going to lose a lot of ions. They're going to lose fluids, okay? Proteins, basically anything that's in the cell is going to leak out if you have a damaged plasma membrane. All right, and also osmosis, you need to learn about it for, most of you are going into some sort of clinical field. Osmosis is something that's very much considered when you're giving someone IV fluids, such as saline, right? You need to know like how much of a concentration of saline to give versus how much water to the person, depending on what kind of situation they're in, right? If they have a deficiency in something, or if they're just low on fluids, you don't want to give them too much sodium, for example. But it's important to understand, what happens, and we'll talk about it. What happens if you're not giving proper concentration consideration when you're giving IV? You can destroy people's cells. All right, we'll mention that later. Osmotic imbalances are also related to symptoms of many different types of diseases or conditions, such as diarrhea, constipation, edema. Edema refers to the accumulation of fluid, and the nature of that fluid really depends, but mostly it's water-based. But it can be filled with other substances besides water. It might be accumulation of adenomous fluid, which is basically inflammatory fluid. But in any event, edema, we're talking about the accumulation of water outside of the cells, and then you end up seeing like a swelling on the outside of the person. Okay, so yeah, osmosis, understanding osmotic imbalances is important clinically. Definition here, what is osmolarity? Osmolarity is related to understanding osmosis. And just understand the basic definition here. So what is osmolarity? It's referring to the concentration of solute. Now again, don't get confused. Osmosis is dealing with the movement of water. But remember, water movement is based on solute concentration. Okay, so you're going to want to look at osmosis in terms of osmolarity. What is the total concentration of solute particles on either side of the membrane? You really want to look at it this way. All right, so now again, here's a definition of osmosis. And the definition here says water moves from areas of low solute to areas of high solute. And that really is the best way to look at it. Now you wouldn't be wrong in saying water moves by osmosis from areas of high water concentration to low water concentration. That's not incorrect, but I am going to discuss it in terms of solute concentration because it's going to be easier for you to define other terms that we're going to talk about in this PowerPoint. Okay, so again, water is going to move towards wherever the highest solute concentration is. Let's look at this top part here. So this is like a beaker or a container of some kind, and there's a little partition here in the middle, this dotted line, and that's representing like a membrane, right? Or if it's a beaker, it's some sort of filter. So on the left side, we're showing solute, okay? Now over here, we also have solute. Now the water, what they should be showing, and it's a little hard to tell from this picture, but yeah, here it is. Okay, so the solute is pictured here in orange. And what they're showing here is that there's more solute on this side, on the left side. There's more solute than there is over on the left side. In fact, I don't see any solute on the left side. All the solute is over here on the right side. The solute could be a protein. It could be an ion. It doesn't matter. Water is going to move towards the area of highest solute concentration. That's what happens. All right. So, and then if you check this out over time, right, what you'll see is that because water is moving from the left to the right, following this area of high solute concentration, obviously, if more water is coming over, you increase the volume, right, on the right side. You're just getting more water. So this leads us to some of these important terms I was talking about, all right? Movement of water is eventually going to cause a pressure, okay? Water causes what's called a hydrostatic pressure. And so this is referring to the pressure of water that is pushing or placing pressure on the plasma membrane. So the more water you get coming in, the more pressure you have on the plasma membrane. Now, here's what you want to try and imagine. And again, relate this to the document I made for you all. Now, let's say water is leaving a cell. Let's say there's a high concentration of a solute on the outside of the cell compared to the inside. If there's a high solute concentration on the outside, water is going to flow to the outside of the cell to meet all of that high solute level. So imagine that if this water continues to leave the cell at an unchecked rate, what will happen to that cell? The cell is going to shrink. It's going to shrivel up. The scientific or formal word for this is called crenation, all right, or to crenate. So that's what will happen. Now look at the other way. What if, let's say, we have a high concentration of solute inside the cell compared to outside? Then what happens? Water from the outside is going to flow to the inside of the cell because it's going to follow where the highest solute concentration is. Imagine it continues on and on. You keep having water entering the cell, eventually that cell is going to fill up with water, and if we don't put this in check, then we have the cell explode. The cell will burst or lyse. Lyse is really the good term for this, all right? Lyse comes from the word lysis, or is related to the word lysis. Remember a term called hydrolysis, all right? So lysis means to break apart, all right? So this cell's just gonna basically bust. It's gonna lyse. All right, so now keep in mind, in both of these cases, well, we get death of the cell. This is why understanding osmosis is important clinically. You need to know, all right, what concentrations of solutes are in solution, okay? So for example, in the bloodstream versus what's in the cell, right? So because in either one of these cases, the cell is going to die, right? So you don't want to overload someone with too much water. You also don't want to overload someone with too much solute, because one of these things may happen and you will end up killing those person's cells. So again, another term here related to all this, tonicity. Tonicity. Tonicity is referring, it's kind of similar to osmolarity. Tonicity is referring to the concentration of solute in the ECF. That is your key. Okay, if you can add this to your knowledge base of all of this, tonicity is referring to the concentration of solute outside the cell, not inside, outside. Okay, keep that in mind. So based on this, okay, there are 3 different kinds or categories of tonicity, which you definitely need to know. For sure we'll have a question about it. You can have an isotonic situation. Iso, the prefix iso means same, all right, or equal, all right? So if the outside of the cell has an equal level of solute compared to the inside, all right, then we say we have an isotonic situation, and the volume of a solution or the cell and inside is not going to change. Basically, in other words, there will not be any movement of water. There will be no need for water to move in that case because we have equal concentrations of solute inside and outside. Hypertonic. Hyper means too much. So if you have a hypertonic solution, now remember when we say solution, we're referring to the outside of the cell. All right. The inside of the cell is not a solution. It's cytoplasm. That's not considered a solution. The fluid situation existing outside of the cell is considered solution. All right. Bloodstream, for example. All right. So hypertonic means you have a high, too much, concentration of solute outside the cell. So this is what you have to put together, all right? If you have a high concentration of solute outside the cell, what is water going to do? All right, if solute's high in the ECF, water from the inside of the cell is going to leave. It's going to go out to the ECF to help balance that high solute concentration existing in the ECF. And again, relate this to something else. If that water continues to go out of the cell will shrink, right? And we have a crenation situation. You really want to be able to put these terms together in a sequence. You'll need to know, first of all, which way is water flowing? Is water going in or out of the cell based on solute concentration? Then you want to imagine to yourself and figure out, does this mean it's a hypertonic or hypotonic situation? All you have to do is see the ECF, is look at how much solute is outside. If there's a higher amount outside, it's hypertonic. And then the other thing you want to do is figure out and think to yourself, well, okay, so water is going to be flowing out in that case. So what's going to happen to the cell? All right. And again, I did detail that out in that document. Hypotonic is the opposite. This means we have hypo means under or not enough. Hypo means we have a low solute concentration in the ECF. So what does that dictate about water behavior? That means water is going to leave the ICF and go inside the cell where there's a higher molarity inside. Imagine what's going to happen. Water keeps entering the cell. This will cause the cell to swell, and we will call this lysine. So what are the effects of, and this is just a picture of a red blood cell in each one of these cases, a erythrocyte. All right, here's hypotonic. Hypotonic, well, first let's look at the middle, sorry. The middle is normal, all right? This is an isotonic situation, and your erythrocytes have this sort of concavity in the middle, this depression in the middle, and that's normal. All right, it makes it perfectly designed for oxygen exchange because all of your oxygen will be out here to the rim of the cell rather than the middle. Anyway, isotonic. Hypotonic means we have a high concentration of solute inside. And otherwise said, we have a low concentration of solute in the ECF. So water is going to go into the cell will get larger, eventually it will burst. On the other side here, we've got the opposite. We have a hypertonic situation. Hypertonic means there's a high solute concentration in the ECF, outside the cell compared to inside. So water is going to leave the cell, And that eventually, if it continues, will cause that cell to shrink, all right, and shrivel, and it will crenate. Here's just another picture of the same thing if you want to look at that again. All right, I'm going to take just like a 1-minute break. I'll be right back. Hey guys, I'm back. So I do think you'll be just fine with osmosis and simple diffusion if you refer to that document I made. I think you can handle any question about that. The third kind of transport here is this carrier-mediated. So this is also a type of excuse me, basically passive transport. So this is when we have a situation where a solute is not able to pass through the membrane by itself. It needs assistance. It's going to need to be carried across. You should know that carrier-mediated transport is very specific. So for example, you can have a transport protein in the membrane. This could be a channel, for example, or what is known as a carrier that will only transport one item. All right, it's specific. So for example, we have glucose transporters on the surface of our cells, and those transporters only transport glucose. They will not transport anything else. All right, so carrier-mediated transport is very specific for what it will transport. Okay, and this is just a little bit here about basically what's going to happen. I mean, basically you're going to have something bind to a receptor site on that carrier. All right, and then remember, these carriers are proteins. They're proteins. And I don't know if you recall from chapter 2 when I was talking about proteins, and I mentioned that when something binds to a protein, it causes that protein to change shape. So because this ligand, it might be glucose, whatever, solute, binds to a portion of that protein, it causes that protein to change shape, which will allow the passage of that solute. Also, something about carrier-mediated transport is that it can be saturated, all right? And that's something you'll also talk about next semester, especially with glucose, is that that these transporters can only carry so many items across at a time, all right? They will eventually get saturated as that solute, whatever they're transporting, as that solute increases in concentration in the area, okay, the rate of transport is going to increase, but it can only go up so high, okay? We call this the transport maximum, okay? Transport maximum is just referring to when that carrier is completely saturated. All right, that's what this is referring to. It's the transporter is at its maximum for carrying anything, and it will not be able to carry anything else across until it's finished transporting whatever is there at the time. Not really a big deal on this. Okay, this is just showing you like a picture of transport maximum, which you can abbreviate as TM. You will not have to calculate this or anything. All I care for you guys right now to know is just the simple definition of transport maximum. Oh, this slide is blurry. We'll get through it. So carrier-mediated transport, you should definitely know. I'm sure you'll have a question of some kind on this. There are 3 kinds of carriers. You have a uniport, a symport, and an antiport. Uni means one. So this particular carrier is only going to transport one type of solute. An example of that is something known as the calcium pump. And we're going to talk about that calcium pump when we do muscle contraction, so don't worry about it yet. Just know uniport means this is a transporter that is only going to transport one type of solute. All right. In this case, the calcium pump only transports calcium. Symport, sim, okay, means together, symporter. So this transporter is going to carry at least two, usually two, but it can do more depending. But typically it's going to be two solutes simultaneously in the same direction. Okay, remember SYM, that prefix means together. So we're going to transport two solutes together in the same direction. This is also called co-transport. All right, when two solutes are transported together, it's called co-transport. Again, next semester you'll look at a few really important examples of these, and one of them is the sodium glucose transporter, which we're not going to talk about in this course. Just know the definition of symport. Antiport. Antiport, we're definitely going to deal with a specific example of an antiporter. An antiporter is a transporter that carries two or more solutes in opposite directions. Anti, that prefix anti means opposite. So we're transporting two solutes in opposite directions. And our very important example is the sodium-potassium pump. We're going to have the movement of sodium in one direction, and we're going to have the potassium simultaneously transported in the opposite direction. It says here 3 mechanisms of carrier-mediated transport. You have facilitated diffusion, which is just what it sounds like. This carrier is going to facilitate or assist some solute from getting to get across that membrane. Primary active transport. Now, primary active transport means this is going to require ATP. Example of a carrier-mediated kind of transport that is also considered primary active transport is the sodium-potassium pump, because it requires energy. Active transport requires energy. Then there's secondary active transport, which we're really not going to get into. It's a little more complicated. Again, you should do that next semester. So facilitated diffusion, remember, what we're doing is we're discussing the 3 ways that carrier-mediated transport is carried out. Okay, don't get confused. At the top here, we said there are 3 types of carriers: uniport, symport, antiport. But there are also 3 mechanisms by which any of these carriers operate. So we're just going to mention a little bit about each mechanism. So the first mechanism is facilitated diffusion. All right, so some molecules, examples are molecules that are maybe more hydrophilic, such as glucose, or maybe they're proteins like amino acids or breakdown products of proteins, amino acids, and then some ions, okay, are not able to cross that membrane by themselves. So they're going to use this mechanism to go down their concentration gradient using facilitated diffusion. There are 2 types of facilitated diffusion. The only difference between them is the structure of that carrier, or of that protein, I should say, that's facilitating the transport of this molecule. You have carrier-mediated facilitated diffusion. Okay, and so these proteins exist in the membrane that are called carriers. And I already described it, actually. So you'll have a solute attached to a site, a binding site on that carrier. The carrier is a protein, so it changes shape. And this is going to cause the solute to be able to be moved to the other side of the membrane. Okay, so it's a carrier. All right, your solute is actually going to bind to it directly, causing that carrier to change its conformation. All right, change its shape. The second type of facilitated diffusion is channel-mediated. And this is a lot, we're going to be dealing mostly here in this class with kind, channel-mediated. Okay, so channel-mediated facilitated diffusion is simply that you have a channel, all right? So you have like this narrow open space in a protein that's embedded in the membrane, and the substance, whatever it is, will move through that channel. Now, I mentioned before that channels can be gated or ungated. Remember that? Channels can have a gate, or they can be open all the time, like leak channels, like potassium leak channels, for example. Okay, so again, this is more just sort of a repeat of what I just said. Carrier-mediated, all right, we're going to have a carrier. Now remember, to be a carrier, it has to be transmembrane. That should make sense. A transmembrane integral protein, recall that transmembrane integral proteins span the entire width of the plasma membrane. All right, you can't have a carrier that's a peripheral protein. How would that be possible, right? The carrier has to span the whole width of the membrane if you expect something to be transported across that membrane, right? So carriers are classified for sure as transmembrane proteins or integral proteins. Again, carriers are very specific for what they transport. And you know, they're going to help assist molecules that maybe are too large to fit through other means. Maybe a molecule is too large to go through a channel. All right, channels are very narrow. Channels are usually going to accommodate for really small items like ions. Okay, but for larger substances, you know, a carrier into it. And an example would be like glucose. Glucose is sort of large. It can't fit through channels, and it can't go through the plasma membrane by itself. So glucose will definitely use carriers. And again, they're going to reiterate, they're going to be very specific. Okay, glucose carriers only carry glucose. And again, this is just repetitive. I've already mentioned all this. Here's the picture of facilitated diffusion. So here is your carrier. All right, we get a glucose carrier, for example, or a glucose transporter. So notice it's a transmembrane integral protein. It must span the entire width. All right, and it is a carrier. So what's going to happen is, let's say this glucose, it's going to come here into this carrier and bind to a specific part of that carrier. When that happens, whenever something binds to a protein, it causes that protein to change shape. Okay, so look over here. It is changing its shape. It's changing its configuration. And this then allows that glucose or whatever to come in. Just another picture of this, another way of looking at it. Same thing. Okay, so active transport. Active transport. There are two kinds. So you can have just regular old active transport, which there are two types, primary and secondary. We are focusing here in this class on primary active transport, not secondary, but you should just know secondary exists. Second type here of active membrane transport is vesicular transport, and we'll just mention a little bit about it. Both of these, okay, require ATP. So energy is needed to carry out the transport through these structures. So these structures, the structures that are involved in active transport and vesicular transport, they're going to be transporting things that maybe are too large to fit through channels. Maybe something that is not lipid soluble, all right, it can't get through the membrane by itself. Or there are special circumstances in physiology where you actually need something to move against its concentration gradient, okay? Or let's say, for example, something is just not able to move down its concentration gradient. For whatever reason, and these are popular reasons, all right, the item cannot get across the membrane with the assistance of energy requiring active transport or vesicular transport. Okay, so let's take the first one, primary active transport. So I sort of hinted at this just a second ago. So primary active transport, sometimes you have special situations, and we're going to see these special situations when we talk about muscle physiology and also when we talk about neurophysiology. There will be special situations where instead of having a solute go down its concentration gradient, so for example, instead of having a solute be in equal concentrations on both sides of the membrane, you may have situations where you don't want that to be the case. In order for some sort of cellular event to take place, you will actually need the opposite to happen. It's actually like a type of positive feedback. Let's say you have a high amount of sodium inside the cell. Well, in primary active transport, we're going to actually continue to make that happen. All right, we're going to bring even more sodium. We're going to keep bringing sodium inside the cell. Rather than balance out the levels of sodium on each side of the membrane, instead of going down a concentration gradient, we're going to go against the concentration gradient. And that is a type of positive feedback. Primary active transport requires ATP. How do we get ATP? Remember, hydrolysis, right? We're going to do hydrolysis of that terminal phosphate on ATP. That is going to release energy. Some examples. Now, don't worry about the details of these right now, okay, except for the bottom one, because we're about to focus on it. Okay, calcium pump is an example of a unipart. We'll talk more about that when we talk about muscle, so don't worry about that right now. Sodium potassium pump, this is the one we worry about right now and all the time for the rest of the class. It is classified as an antiporter or an antiport, which means we're going to be simultaneously transporting two different entities in opposite directions. All right, and what we're transporting is sodium one way and potassium the other way. So here it is, sodium potassium pump. And I do have another short 7-slide lecture I'm going to give after this that details out that sodium potassium pump. Sodium potassium pump is absolutely critical in physiology. You cannot live without this. Pretty much every cell in the body has a sodium potassium pump. So much of the cellular action that goes on inside of a cell, the foundation of whether or not those activities can occur is based on the proper functioning of this sodium potassium pump. Absolutely critical for life, okay? So yeah, we're going to talk about it. The sodium potassium pump is an example of primary active transport. And it is an enzyme, okay? It is an enzyme. So because of that, we can also add our ending here of ASE. You can also call it the sodium potassium ATPase. All right, so this is a protein which you can classify as an enzyme. Really critical to know. I mean, you will have 2 or 3 questions about the sodium potassium pump, so you definitely want to be familiar with it. So what this pump or what this ATPase does is it's going to pump out 3 sodium ions. It's going to send out outside the cell. It will pump 3 sodium out per every 2 potassium that it brings into the cell. Remember, antiporter. So we've got 2 different substances going in 2 different directions. You must know the numbers here. You must know the numbers here. It's absolutely critical in understanding these numbers and which ions are involved and which way they're going. Must know it, gotta know it. 3 sodium out, potassium brought in. Now, it says here, yeah, sodium potassium pump is on all plasma membranes. Remember, this is on the surface of all of your cells, but it is really, really abundant on our excitable cells. And that's because, you know, in order for our nerves, I should rather say neurons there. I'm not sure why I have nerves there. Neurons and muscle cells, okay, have to have a properly functioning sodium potassium pump, and they're going to be all over the place. The sodium potassium pump is going to be all over the place on the plasma membranes of these excitable cells. Okay. Now remember, this is primary active transport. So what we're doing here, The sodium-potassium pump does not function based on simple diffusion. It's not related to that. This is primary active, totally different kind of transport. So what that means is we are not having these ions go down their concentration gradient. It's the opposite. We are going to be sending sodium out and potassium in against their concentration gradients. And that probably doesn't make a whole lot of sense right now, but it will make sense later, hopefully. All right. So because we're going against the concentration gradient with the movement of these two ions, energy is required for that. All right. So that's why we have to have ATP there. Energy is needed for that process. I hate when these slides do this. So this is more about the sodium-potassium pump. And the takeaway from this slide, I can't really read it, but I can tell you that the take-home message about this slide is, okay, what is the sodium-potassium doing? Okay. First of all, we're going to use an ATP molecule. We're going to hydrolyze an ATP molecule in order to cause the exchange of sodium for potassium. Now, here's what the deal is that the cell, in its natural relaxed state, is going to have a higher potassium concentration on the inside of the cell. The rest. Okay? A normal relaxed cell, when it's not excited or stimulated by something, when it's relaxed, it has a high concentration of potassium inside the cell compared to outside. Sodium is the opposite. In the relaxed state, there is a lower concentration of sodium inside the cell versus the outside. And the reason we're able to maintain this situation is because of the sodium-potassium pump. Think about it. What is the potassium pump doing? It's pumping out more sodium than it is pumping in potassium. It is sending out 3 sodium, which is what helps keep the lower concentration of sodium inside the cell, because we're getting rid of 3 sodium, getting rid of it. We're sending 3 sodium out of the cell. So that helps maintain this relaxed state characterized by a lower concentration of sodium inside the cell. Look at potassium. Okay. Now, potassium at rest, there should be a higher concentration of potassium inside the cell. So that is maintained, that lower concentration of potassium inside is maintained by the fact that the sodium-potassium pump is bringing in to potassium. Okay? So this pump maintains the cell at rest. It helps keep your cell in homeostasis, all right? Because sodium and potassium are so absolutely critical for so many cellular events that take place. So this pump keeps the cell in its sort of default or relaxed state, all right? By getting rid of more sodium, keeping sodium in the ICF low, and bringing in to potassium, helping to keep the potassium ICF higher. Okay, so that really is the main idea of this. Okay, now I do want to point out something down here that relates to leak channels. All right, so before I mentioned the different, we have gated channels, non-gated channels. Leak channels. So leak channels are channels that are always open. The most important one in physiology is called the potassium leak channel. And so what's happening in cells is that potassium is it's constantly leaking out of your cells at a slow rate. Okay? Potassium leak channels, we're having potassium leave the cell at a slow rate continually because this channel is always open. So think again what this sodium-potassium pump is doing. It's helping to counteract or assist with, in the event that you have too much potassium leaving. It's sort of counteracting that a little bit, right? By bringing back in some of that potassium. Here is the sodium-potassium pump. What you're seeing here is, okay, so in blue is potassium, in orange is sodium. Now remember, the inside of the cell has a higher concentration of potassium compared to sodium. And you can see that. There's more little blue dots on the inside. Okay? Now, there are fewer sodium ions inside. That is the normal relaxed state. And the reason we have fewer sodium ions inside the cell is because of the sodium-potassium pump. It is pumping out. It is taking out 3 sodium ions at a time. And so for every 3 sodium ions it pumps out, it's going to bring back in 2 potassium ions. And again, the fact that it's bringing in these 2 potassium ions contributes to why there is a higher concentration of potassium inside the cell compared to out. And that is the normal relaxed state. Notice that in order for this pump to operate, in order for us to be able to have this action of pumping this sodium out, first of all, you have to have hydrolysis of ATP. Okay? We need energy to do that. All right, like I said, I do have a short lecture going through the specific mechanics of the sodium-potassium pump, and I will put that on here just after I'm finished with this one. Okay, so we're getting there. Vesicular diffusion, we're still on this, and actually this is a repetitive slide. We've already talked about this. Channels, okay, so channels, remember we said there are carriers and channels. Channels are transmembrane proteins, and they're kind of like carriers, but they have a different structure. They have a narrow opening, all right? And sort of similar to carriers, they are going to be pretty specific for what they transport. And remember, one example of a channel is an aquaporin, and it's going to be specific for the transport of water. 2 types of channels 2 main types, leakage. Leakage channels are always open. I just mentioned the most famous type of leakage channel, and that's the potassium leak channel. Gated channels are controlled. They're going to open or close based on some sort of chemical or electrical signal. We did talk about that in chapter 2, or maybe the first part of this chapter. So, and here's just a picture again of channel-mediated facilitated diffusion. So here we have this narrow opening, all right? Again, the channel is different from a carrier in the sense that in a channel, you know, the solute is just going to go straight through, right? If it's like a leak channel, for example. If it's a gated channel, it'll need to bind to the gate, and the gate will open and it will come through. But basically in channels, you don't need to have the solute actually bind specific to the channel. The channel does not need to change conformation. It doesn't need to change shape like we saw with carriers. All right, so that takes us to this other kind of transport that I'm not going to dwell on too long. All right, I really think that you just need to know the basic definitions of these. Okay, so your type of active transport is called vesicular transport. What we're doing in this case is we're transporting large things. All right, large things that they're large enough that we're going to actually need to encircle those things into little sacs. These little sacs are called vesicles, so that's why it's called vesicular transport. Remember, this is a type of transport, so it does require ATP. All right, now there are 2 main types of vesicular transport. First, you have endocytosis, and then you have exocytosis. Endo, meaning into or inner. So endocytosis, remember cyto means cell. What we're trying to do is bring something into the cell. All right. What is that? Okay. I'm sorry. I should have had my slides full frame the whole time. All right. So yeah, endocytosis, we're bringing something into the cell, something large. Now, there are 3 different kinds of endocytosis, but it's really pretty straightforward, okay? The first kind of endocytosis is called phagocytosis. Phago means to eat, to eat. So phagocytosis, we're bringing in some kind of solute into the cell. Now remember, this is going to be a large solute that is going to need to be placed into a vesicle. All right, this is not going to go through a channel. This solute is not going to go through a carrier or a pump. It's going to need to be enveloped by the plasma membrane. The plasma membrane is going to reach out basically and envelop this large solute, place it in a vesicle, and then the solute will be able to travel around the cell. Pinocytosis. Pino means to drink. So here we're going to take in some sort of a fluid. The third type we are not going to deal with. It's much more complicated, but you know it exists. It's called receptor-mediated endocytosis. So basically something is going to bind to a receptor on the plasma membrane, and then the plasma membrane, again, will envelop that and bring it in. This is a really detailed and specific one that we're really not going to go into. So exocytosis is simply when we have the transporting of something large out of the cell. Exo, exit. We're going to have something exit the cell. So we'll go through each of these hopefully kind of briefly. So the first one, phagocytosis. All right, what we're going to have happen is the membrane, plasma membrane, will have these little will form something called pseudopods. You've heard that word before, false feet. Okay, so the plasma membrane will actually sort of extend out a little bit, forming these arm-like structures, or foot-like structures. And this will envelop or engulf whatever is going to be taken in. Okay, and it brings that substance in, and then the membrane itself is going to pinch off. It's going to pinch off, forming a vesicle. All right, this vesicle is called a phagosome. Now, you should know that phagocytosis is really popular in macrophages. And I think I introduced that cell to you before, the macrophage of the immune system, and some other types of white blood cells that have these pseudopods on them. And they're going to do phagocytosis. Here's just a picture of phagocytosis. So let's say this is some object, something that's being brought into the cell for whatever reason. Now, this purple lining here is the plasma membrane, all right? And the little light, the other little purple structures are little receptors. So, but look how the plasma membrane sort of has these pseudopods on it, all right? These arm-like structures, as if the plasma membrane is enveloping and engulfing something. And so it brings this in. And what's going to happen is that these two pseudopods, once we start to bring this substance in, pseudopods are going to end up attaching together. They're going to fuse, all right? They're going to fuse together here at the top. And that is going to cause this vesicle that has now been formed around this substance to pinch off from the membrane. All right, pinocytosis is just like phagocytosis. It's just that we're bringing in a fluid instead of a solid. This is really, you see a lot of this happening in absorption in the small intestines, because by the time your food gets to the small intestines, you know, it has been pretty much digested down all the way. So most of your nutrients at this point are in a fluid state. So your little microvilli down there are going to be doing absorption using this method of pinocytosis. Okay, so a picture of pinocytosis really is just like, or not, I shouldn't say just like, but it's very similar to phagocytosis. Here we're just having a fluid that's being sort of enveloped by the plasma membrane, and it ends up forming a vesicle that will pinch off from the plasma membrane. And then that vesicle will be transported all around the cell to wherever it needs to go. So exocytosis is the opposite. We're going to be ejecting materials from the cell. Okay. Now, before it gets ejected, it is also enclosed in a vesicle. All right, we call this a secretory vesicle because eventually that vesicle is going to secrete something out of the cell. Now, a couple of little terms here we should know. So exocytosis is actually a really complicated and detailed process that we don't need to get into the detail of. Any of you going into pharmacology, if you are, will definitely be studying this in more detail because a lot of drugs target this process. All right, you can take drugs that inhibit the secretion of certain items, for example. All right, so anyway, on this secretory vesicle that's existing in the cell that contains some kind of material that we want to get rid of, okay, on that vesicle, there is a protein on the surface of that vesicle, and it's called a V snare. V stands for vesicle in this case. Now, go to the plasma membrane. On the plasma membrane, we have a different protein called a T snare. T stands for target. So what we're trying to have happen here is we need to have this vesicle, which is somewhere inside the cell. Well, it doesn't know how to get out of the cell. I mean, if it didn't have these proteins, it wouldn't. Here's what happens. The vesicle with its V snare on the surface is going to find a target on the plasma membrane. Okay, the T snare is a protein there on the plasma membrane. And the V snare, V for vesicle, is going to join up with the T snare, T for target, on the plasma membrane. And it's like a docking process. It is called docking, kind of like docking a boat, right? You're bringing your boat up to the dock, okay? And you're going to tie off there, all right? The V snare needs to connect to the T snare. Once this happens, we can get exocytosis. And, you know, like just examples, what kinds of substances will be exocytosed? Hormones, of course, because we make hormones inside of our cells, and we need to send them through the bloodstream. So hormones will definitely be exocytosed. Same with neurotransmitters. Neurotransmitters are the chemical signals of the brain. Mucus, which we've mentioned mucus in lab, and then waste, of course. And these are just examples. So here's a picture of a example of exocytosis. Here is a secretory vesicle containing some sort of material. And here are these linking proteins. Okay, the linking proteins meaning the T snare and V snare. They're going to connect. All right, so that this vesicle can dock with the plasma membrane. And once that docking occurs, the vesicle membrane fuses with the cell membrane. All right, they become one. And now that material can be released. And really a neat little picture over here of this actually happening. Now, here is a picture of this whole docking process, and it's not nearly as complicated as it looks, okay? So take up here, the top one, we have a secretory vesicle containing something that we need to exocytose. And notice that on the surface of this secretory vesicle, we have a V snare, V as in vesicle. Okay, that is on the surface of the vesicle. And so this vesicle is going to go around, and these V snares are going to search out a target. All right, it's going to search out a T snare. So T snare is a protein on the membrane of the cell. All right, so the V snare will connect with the T snare. And that's what's happening in this second picture. You see how the two proteins, the V and the T snares, join, they fuse. And then down here you see what starts to happen then. Okay, now the vesicle membrane is now going to fuse with the plasma membrane, and now we're able to release the contents of the vesicle. this I'm not going to ask you details about at all. Okay, this is just, excuse me, an example of a process where you can have phagocytosis, something's brought in, and then you have like a cycle of digestion of whatever that is that was brought in. And then you might have that product released again in a different form. Okay, but again, I'm not going to ask you details about that. That is it for part 1 of chapter 3. Next, I am going to be posting a short, very short video about the sodium potassium pump. All right, so I'll see you all later. Thank you.